So welcome everybody to the next installment of our Translational Science Seminar Series. We're going to hear from Dr. Samantha Zambudo today. Prior to talking about her work, I do want to go over a couple housekeeping items related to the CCTS Spring Conference. Because of some of the weather issues that we had, we've extended a few deadlines. So remind everyone that the abstracts are now due this Friday, the 6th of February. And anyone that is preparing a mentor nomination, we need those submitted by the 12th of February. If you have any questions, feel free to reach out to myself or Vicki King. We can get you taken care of with regards to the conference. So, the star of the show today, Dr. Samantha Zambudo, is an assistant professor in the Department of Mechanical and Aerospace Engineering here at UK. Her research focuses on developing innovative technologies through biomaterials engineering with a particular focus on women's health. And that is all I'm going to say. I know I've seen the first couple slides, and Dr. Zambudo is going to do a wonderful job of introducing her path and her work. And so I'm not going to take any more time. The floor is yours.
Great. Thank you so much for the introduction. I'm just double checking that the mic is working and you all can hear me and see the slides. Okay, great. So thank you all for being here today to listen to my seminar. I'm going to start by sharing a little bit about my background and how I ended up here at Kentucky as well as in this field. So I'm classically trained in biomedical engineering through and through, starting at the bachelor's level and going up through PhD level. And I also did a Master's of Population Health Science degree because I'm very interested in public health, as well as the intersections of um, public health and how we can use that through our development of engineering models. And then in 2025, I came to the University of Kentucky and started my lab in the Department of Mechanical and Aerospace Engineering. So, a common theme amongst all of the different experiences that I've had in research really revolve around the concepts of hormones and health disparities. I have worked in a variety of different labs. I started off as an undergrad doing research in molecular cloning and protein purification. I spent a lot of time working with E. coli and getting them to express different proteins and then isolating them. From that experience, I really wanted to get into tissue engineering, which I will talk about later in this talk. But I was very passionate about tissue engineering, so I ended up pursuing a master's degree at Brown University. And I worked in a neuroscience and tissue engineering lab, and I looked at sex differences in stroke and traumatic brain injury. And for me, this was the first time that I started thinking about health disparities in science and how we can use tissue engineering models to improve healthcare for all people. So for me, this started out with looking at sex differences, but from this experience, I became very passionate about women's health and wanting to study women and improve health and science research for women in particular. So that led me to the University of Illinois at Urbana-Champaign, where I did my PhD, and I focused on developing biomaterial-based models of early pregnancy. And then after my PhD, I did a postdoc at Washington University in St. Louis and started studying aspects of vaginal biomechanics. And then after all of that, in January 2025, I started my laboratory here at the University of Kentucky. And we are a tissue engineering lab that focuses on women's health. So in our lab, we utilize multidisciplinary approaches to solve problems in women's health unified through engineering innovations. And today I'll spend a lot of my time during this talk sharing our technology with you all and how we approach using engineering techniques to study various aspects of women's health. So as I mentioned before, I'm really passionate and interested about tissue engineering and women's health research. And so in my lab, what we do is we create these tissue engineering systems of the female reproductive system as well as other female specific tissues. So if you know anything about the tissue engineering triad, what we do at the most foundational base level is we combine biomaterial scaffolds with cells and we give them some kind of signal to create engineering systems. And we've created systems that span from maternal fetal interface to tissues in the female reproductive system. And now we've moved into creating tissue engineered systems of the bladder as well. And then when we develop these types of systems, science and translational applications. So really that means that we're using these types of model systems to understand various aspects of female reproductive health and women's health generally, and then also using them for translational science purposes, which I will also discuss later in my talk. So you may have noticed that my lab is fully an in vitro system lab. We build these tissue engineering systems and we use them with cells. So you might be wondering, well, why tissue engineering models? Why should we care about these types of in vitro systems? And the reason for this is a few different reasons. So first, I'd like to bring up the FDA Modernization Act 2.0, which was passed in 2022. But we have seen some interest in these engineered systems that have been becoming popular over the course of the last few years. Generally, engineered systems have lower cost and they're also much more high throughput when you compare them to animal testing. And as I will talk about with 3D systems in particular, they replicate more of the native anatomy and physiology of tissue systems. And importantly, though, these types of bioengineered and engineered systems have actually started becoming a part of the drug development pipeline. Um, so in this image here, and I think you can all see my mouse, but so this part here, this top part of this image shows the traditional pipeline for drug discovery. So typically you would discover some kind of molecule for whatever application you are looking at. And then traditionally you would use preclinical tests to demonstrate safety and efficacy. So that would typically be with in vitro testing in cells, which traditionally are cells grown on plates. Then, once you proved that it was efficacious for that, then you would move into animal testing, and then subsequently from animals, move into clinical trials. Um, the way that the FDA Modernization Act 2.0 changed this drug development pipeline was that they changed what we refer to as preclinical tests to non-clinical tests. And so what non-clinical tests they're defined as these tests conducted in vitro, in silico, in chemico, or a non-human in vivo test. So instead of having this, traditional start with a 2D cell culture model, move into animals, and then into clinical trials, we're seeing that all of these types of systems together are now what are referred to as non-clinical tests and what you would test drugs in prior to moving into humans. And you'll notice a lot of these systems are traditional engineering systems like organ chips, tissue engineering models, in silico models like digital twins, and organoids as well. Animal models are still here, but we're also seeing this increase in popularity of these engineering systems. And the reason for this in particular has to do with significant translation failure rates, especially if you think of cases where animal models might not be the most robust systems for testing drugs and efficacy. So in particular, that's things like asthma, inflammation, or probably one of the more well-known cases is with neurodegenerative diseases, such as Alzheimer's disease, where you see a 99% translational failure rate between testing in animals to testing in humans. Um, so we're still ultimately seeing how this will change the paradigm of the drug development pipeline and how people will continue to use these types of engineered systems. But the main goal of this act in particular was to try to improve this drug development pipeline in the case where we have significant translational failure rates. But nonetheless, this brings up an important consideration that if we are going to be using these types of engineered systems in studying drug safety and efficacy, we need to create more accurate engineered systems to make sure that we are actually demonstrating safety and efficacy of drugs in particular. And I'll talk a little bit more about some examples in women's health later on and how this relates to that. Another important reason why tissue engineered models are becoming popular and relevant in science also relates to funding priorities. In particular, in 2025, the NIH made this announcement where they wanted to prioritize human-based research. So they refer to these types of non-clinical bioengineered model systems as new approach methodologies. So again, these cover things from organ chips to tissue engineering models, as well as digital twins, et cetera. And their goal for announcing this is that so they're no longer exclusively supporting the use of animal models. So they really want to try to get people to focus more on the development of systems that are using human cells, with the hopes that they more accurately represent human physiology and anatomy. Um, so we're seeing more of these types of systems being prioritized, not only in terms of thinking about applications in drug discovery, but also basic science approaches as well. So that's a little bit about the background about tissue engineering models and why we generally care about them. Um, but the fundamental goal of my lab's work is really to eventually take these engineering tools and be able to bring them from bench to bedside to improve patient care. So I'm really grateful to the CCTS here at UK for generously awarding me pilot funding as well as KL2 scholar support for my work. So my work has a very heavy focus now on translational science. So yes, we're developing these models to understand basic aspects of human physiology as well, but the ultimate goal is to be able to adapt these types of engineering systems and use them, for example, for precision medicine purposes. And so uh, the National Center for Advancing Translational Sciences defines translational science as the field that generates innovations that overcome longstanding challenges along the translational research pipeline. And as we talked about with the FDA Modernization Act 2.0, as well as the NIH push to use new approach methodologies, the goal of my lab really fits in the historical context right now of science. And our goal is really to develop these improved tissue engineering models to directly impact human health and disease. And the types of models that we work on in the lab that we will talk about later in the talk really focus on a variety of core principles of translational science. So for example, we prioritize initiatives that address unmet needs in particular for women's health generally, as well as understudied applications in women's health. And our goal is to produce these generalizable solutions via engineering tech. So I will focus on particular tissue and organ systems, but just know that the base principles of this technology have the opportunity to be applied to other types of systems as well. And then we also work using a lot of creative and innovative techniques, and we really prioritize interdisciplinary teams in our collaborations as we develop these types of systems. So for today, I've divided my talk into three different sections. We will first talk about biomaterials and the types of systems that I use in my lab and why we think they work very well for these types of models and applications. Secondly, we'll talk about model development and how we use engineering principles to design these types of tissue engineered systems. And I'll share a couple of vignettes of our previous data working with development of these systems. And then finally, I will end my talk with a brief discussion on applications and what we are doing with these types of models and what we plan to do with them as well. So to start, I will start talking about biomaterials and the biomaterials that we use in our lab. So our lab revolves around the use of these biomaterial systems, and at the fundamental base level, biomaterials are any materials that interact with biological systems. And for us, we work with hydrogels. Hydrogels are cross-linked polymer chains, and they can absorb large amounts of water. They're very widely used in tissue engineering. There's a lot of different materials you can use to make hydrogels and a lot of different manufacturing approaches you can use to create these types of systems and change not only their mechanical and material properties, but things like their shape as well as how you would apply them in particular. Um, And generally, hydrogels are really great for tissue engineering in general, since they're biocompatible, biomimetic, and they can support three-dimensional cell culture, which, as we'll talk about, is relevant to replicating aspects of native tissue and organs. One of the most commonly used biomaterials for tissue engineering, as well as stem cell biology, is Matrigel. People typically like to use Matrigel because it's an excellent, easy to use substrate. It works really well, cells like it, especially stem cells, but it does have its issues. For example, batch to batch variability, heterogeneous material properties, and the lack of controllability in terms of the actual engineering of the system. So as we think about ideal types of biomaterials to use for tissue engineering, we want to be able to replicate a lot of these different features that I've just mentioned. So with biomaterial science, you can begin to create biomaterials that are reproducible, consistent, as well as highly tunable for whatever application that you are using. So in our lab, we use a material called gelatin methacrylate or GelMA. This is a really great material that we have used as well as others, and not just for women's health applications, but for a wide variety of other tissue and organ systems. So GelMA is derived from gelatin, which is denatured collagen. So gelatin itself maintains these relevant RGD cell binding motifs as well as MMP sensitive degradation sites. So what this means is that cells can readily bind to the material as well as degrade it and remodel it as they're beginning to interact with the material. But if you know anything about gelatin itself, whether you've worked with gelatin or maybe you remember making Jell-O back in the day, gelatin itself is not stable at physiological temperatures, which means to use this material, you need to use uh, biomaterial science and chemistry approaches to modify it so that you can use it at physiological temperatures. So this is essentially what we do in our lab. We synthesize GelMA through chemical reactions using methacrylic anhydride. And what this ultimately allows us to do is to create this GelMA material that is not only stable at physiological temperatures, but because it is photopolymerizable via the use of a photoinitiator and light, in our case UV light, you can create these cross-linked hydrogels with varying parameters that you can choose. So compared to other types of materials, there's a lot more parameters that you're able to tune in regards to this material in particular. So you can work with different types of gelatin to make different variants of GelMA. You can modify the degree of functionalization of your GelMA backbone, which enables you to create more or less cross-links. And there's a wide variety of parameters associated with hydrogels themselves, such as the one shown here, that you can use to create different types of geometry, patterning, stiffness, etc. So you can really take these materials and create these types of systems that are very highly relevant to the native ECM within the body. And so this is an example of how we use GelMA in our lab. We have these 5 mm Teflon molds, and we create these three-dimensional cylindrical hydrogels. They're pretty small, so they're relatively high throughput. And we've also done a wide variety of characterization over the years for these types of materials, and we've shown that their stiffness and their properties are relevant to a wide variety of tissue systems. So for this batch that I'm showing in particular on the right, the stiffness is approximately 100 pascals, so very soft for these materials. And for these ones in particular, we were using them to study the endometrium, which is a very soft material, which has stiffness of roughly 100 pascals as well. So they are very highly relevant to the mechanical of the tissue systems that we are interested in studying. So that was a little bit of background on the materials we use. So now I'm going to discuss how we use these biomaterials to create engineered systems. And so we've predominantly spent a lot of our time working on creating these tissue engineered models of the female reproductive system. We've created models of the maternal-fetal interface to study aspects of early pregnancy. We've created models of the endometrium, again, in relation to being interested in studying pregnancy. And we've also worked in the space of creating models, tissue-engineered models of the vagina for studying biomechanics. So today I will share two different vignettes. I'll start by talking about our model systems on the maternal-fetal interface and the endometrium, and I'll share some model highlights and how we went about using these types of model systems. And so going back to what I alluded to at the beginning of my talk, in certain cases, animal models are not necessarily the most robust systems for studying human physiology. So when we think about human pregnancy, it's pretty unique to humans in particular. And although animal models have been useful in understanding human pregnancy, they are again not always the best, and sometimes they can't recapitulate aspects of human physiology in this case. So our motivation for the creation of these models of early pregnancy was that we really needed to create better models specifically of human pregnancy. And if you look at this image here, you can appreciate that the uterine anatomy is very distinct between animals and humans, as well as placental anatomy as well. And also the bipedal versus quadruped anatomy of humans compared to animals can affect the biomechanics of these types of systems. And a real tragic event in history that is a reminder of how animal models might not necessarily be the best models for pregnancy relates to the thalidomide scandal of 1961. So if you've heard of this, thalidomide was a drug that was prescribed early in pregnancy to women to treat morning sickness. It was extensively tested in animals, such as all of those different animals shown on this slide. And they demonstrated that thalidomide had no significant teratogenicity in these animals when they were given the drug during pregnancy. However, tragically, when women took this drug when they were pregnant early in the first trimester, it unfortunately resulted in very severe birth defects. So these are pictures of two children who are suffering from phocomelia, which is a congenital abnormality which affects the limbs. The thalidomide crisis affected roughly 10 to 20,000 infants, but many died at birth, unfortunately. So this is a tragic reminder that, with animal models, we do our best um, to consider the safety and efficacy of drugs, but sometimes they're not enough and they're insufficient for actually capturing what would happen in human beings. So again, it really begs the question of, how can we create better model systems to study drugs with human cells? And how can we improve these systems to be able to use them for situations like this, where we want to examine, the safety or efficacy of a drug for pregnancy in particular. So for us, again, we're developing these models to understand mechanisms in this case, but also for translational purposes, because eventually we could take these models and move them into precision medicine and drug testing for patients. So as we thought about models of pregnancy in the endometrium, we came up with a series of engineering criteria for developing these types of model systems. So for us, what we do is we think about the context of this tissue and we think about what might be the most relevant series of criteria to begin to model in our systems that can replicate relevant features of the tissue that likely have an importance in how the tissue functions. Um, so again, the endometrium is the lining of the uterus, as well as the site of blastocyst implantation during pregnancy. This image on the slide here shows the endometrium over the course of an entire menstrual cycle. So over the course of a month, starting at menstruation and working its way through the secretory phase. Um, the During the menstrual cycle, this tissue is basically preparing for the potential of pregnancy. So basically, it goes through a series of differentiation processes that are orchestrated by steroidal sex hormones. and ultimately leading up to the secretory phase of the menstrual cycle, which leads to this receptive window where a potential blastocyst could implant and establish pregnancy. And then this process is non-reversible. So if pregnancy is not established by the end of this process, the tissue is then shed during during menstruation, and then the process begins again the next month. You can imagine that as this tissue goes through this complicated differentiation process, it really requires a really complex spatiotemporal dialogue between cells on the maternal side within the endometrium, as well as cells from the blastocyst side, which are trophoblast cells, which we'll talk about later. But it really requires this complicated dialogue to establish and maintain pregnancy. So that being said, and with these core features of the tissue in mind, we defined the following engineering criteria. And so again, we wanted to replicate the overall structure of the endometrium. So if you're looking at this picture, you could see that it has an epithelial layer that's overlaying a stromal vasculature compartment. So that was one consideration that we thought about. We also wanted to be able to replicate the differentiation process that occurs during the menstrual cycle, especially being able to replicate these aspects of receptivity and the tissue preparation for pregnancy. And then we wanted to make sure that we were incorporating more than one cell type to be able to ensure that we're replicating, this stromal vasculature compartment in the tissue as well as the epithelial cells on top. And then finally, incorporating aspects, which we'll talk about, of maternal and fetal cues to replicate these aspects of early pregnancy. So these were the first set of criteria that we defined as our engineering criteria. But the nice thing about these types of model systems, and as I'll highlight, are that we can actually use a modular approach to the development of these systems. So we can increase our complexity as we continue to create, develop, and optimize these model systems. And we can strive toward creating much more complex systems that are more representative of human physiology. So this is what it looks like in cartoon form if you're thinking about, taking a snapshot of what the endometrium tissue would look like. It has an epithelium overlaying a stromal vasculature compartment with hormonally stimulated endometrial stromal cells, as well as cells from blood vessels. So we approached this in a modular way where we optimized creation of an overlaid epithelium on our hydrogels to replicate this part of the tissue. And then subsequently, we created hormone responsive microvascular network system in our hydrogels to replicate this aspect and replicate the endometrial vasculature system. And then we subsequently combined both of these systems together to create one triculture model of the endometrium. So if you remember from early in my talk, I mentioned that a benefit to using GelMA as the biomaterial system is that it contains relevant cell binding motifs, so cells can readily attach to the gel in particular. So what we did was we took primary human endometrial epithelial cells and basically cultured them on top of our hydrogels as a simple endometrial epithelial cell model system. And so what we found was that within 3 days of adding the cells onto our hydrogels, we were able to see that the cells had an epithelial phenotype where you're seeing cytokeratin 18 expression toward the outsides of the cells, which is what you would also see. But if you look more closely at this image too, you're still seeing that we're not getting a perfect monolayer. We were still getting some gaps between the cells. And for us, it was important to be able to create a full monolayer, especially as we were interested in studying cell-cell interactions. We wanted to make sure that if we're adding other cell types in here, they're interacting explicitly with the cells and not just the hydrogel. So what we did was we collaborated with a group at the University of Illinois Urbana-Champaign, which was led by Dr. Gregory Underhill. And in his group, they used these microarray systems, which are another type of engineering system. And what they can do is they take a polyacrylamide gel and they can array different ECM combinations onto these gels and then seed cells on top. So what this allows you to do is a rapid screen of, in our case, 55 different combinations of ECM biomolecules, and then put cells on them and see, which ECM combinations best allow for cellular attachment. So in particular, what we did was we again took our epithelial cells and then we identified 10 different ECM biomolecules present in the endometrium and seeded our cells on those single and pairwise combinations of those ECM biomolecules. And then we looked at cellular attachment over time to see which of these ECM biomolecules might help facilitate epithelial cell attachment. And the reason for doing this is that, well, if we can't add the cells directly onto the hydrogels without getting a nice monolayer, if we add an ECM layer underneath them, in between the cells and the hydrogel, we can likely facilitate better cellular attachment. And that's exactly what we did. So we counted the cells per island and then generated this heat map, which shows that based on the different combinations, single and pairwise, of the ECMs, we get variable cell attachment depending on what ECM the cells were attached to. And then we found that the best attachment of our epithelial cells related to combinations of collagen 1 and 3 or collagen 4 and tenascin C. So then taking this information, what we can do is then test it back in the hydrogels to improve our model system. So we use a technique using microbial transglutaminase. What this technique does is it allows you to enzymatically cross-link different ECM biomolecules onto the hydrogel surface. So this is just an example showing that we're able to tether molecules such as laminin onto the hydrogel surface. So instead of just a plain hydrogel, you now have a coated hydrogel system. And then what we did was we had our control, which was an uncoated Gelame hydrogel. We took typical ECM coatings that people use in the literature to adhere cells better to hydrogels, and then our microarray hits. And we demonstrated that we're able to get better cell attachment with these different combinations of ECM biomolecules compared to that of what we had previously seen in the literature. And stay tuned, we'll talk about how we assembled the triculture after. But the next thing I will highlight from these model systems was that we were very interested in creating microvascular networks in our hydrogel systems to replicate the vasculature in the endometrium. So what we do is create co-cultures within the hydrogels of endothelial and stromal cells. If you grow endothelial cells alone, typically they fall apart within hours, especially in systems like Matrigel. You will get endothelial tubes for hours, and then they will eventually break down and fall apart. But if you combine them with a support cell, such as stromal cells, you can get longer term culture of these types of microvascular networks. So for us, we wanted to see how far we can go, because if you remember from my previous slides, 28 days is the average length of a menstrual cycle. So we wanted to see if that was something we could do. So we used a custom MATLAB algorithm to determine the total network length, total number of branches, branch length, and total vessels in our hydrogels, in our hydrogel cultures. And we looked at this for 7 to 28 days. And we tested varying ratios of the endothelial to stromal cells to see which ratio would be the best and the most optimal one for our case. So unfortunately, with the 1-to-1 ratio of endothelial to stromal cells, this was the least stable. And by day 21, the cells had completely degraded the hydrogel system, so we weren't able to use these. The 1-to-2 endothelial to stromal ratio cell were a bit more stable than that, and they were able to grow out to 21 days. But you could see that by day 21, we started seeing a crash in our system where the total network length and number of branches, as well as total vessels, began to decrease over time. And then finally, with the 2-to-1 ratio, this was the most stable ratio of endothelial to stromal cells over time that allowed us to culture our cells for 21 days without seeing this crash in the vessel networks. And we did attempt to go to 28 days, but after day 21, the cells had completely degraded the matrix. Um, but still, 21 days was quite a long time to be able to culture these cells in vitro to perform in vitro studies. The next thing we wanted to do was, after we optimized our microvascular vessel network model, was to see if we could provide hormones to these microvascular networks to replicate a differentiation process within the stromal cells. So we took our microvascular networks, we added hormones, and then we did a cytokine array to verify whether or not the cells were demonstrating uh, the differentiation process that we were looking for. And so typically in the literature, you look for increased secretion of prolactin and IGF-BP1 by these stromal cells when they are given hormones. And so we tested 2 different concentrations and formulations of hormones that people commonly use in the literature for this differentiation process. And we were able to demonstrate that um, as you add hormones to these systems, you do see the characteristic increases of prolactin and IGF-BP1, which indicates that we are able to differentiate our stromal cells in these model systems with the endothelial cells as well. So this represents our ability to create tissue engineered model systems that are hormone responsive in this case. So finally, taking all of these systems together, we were able to create our triculture model with our epithelial cells growing on top, in particular using this our gel coating that we previously determined, and our endometrial microvascular network models. And we're able to get much better attachment of the epithelial cells on top of the hydrogels, and we're able to create this triculture system. And so just to reiterate the highlights of this model system, again, with our GelMA system, we are able to create these microvascular cultures that are over 20 days, that are able to last over 20 days in culture. We can use hormones to simulate this hormonal differentiation that the cells go through, and we were able to create a stratified triculture model. So we have a triculture model of the endometrium that we can then use for a variety of different applications. So to conclude this vignette, I will also highlight the engineered model systems that we have come up with in order to replicate aspects of implantation. So again, we were interested in early pregnancy, so the blastocyst stage. So at this point in pregnancy, prior to becoming an embryo, you have the blastocyst, which is the inner cell mass shown in pink and yellow, and it's surrounded by trophoblast cells. And these trophoblast cells are ultimately what will attach to and implant into the endometrium to establish pregnancy. So we were really interested in figuring out a way that we can create these, like an engineered system to study these trophoblast cells in particular, especially in early pregnancy. And so what we were able to do was we used the Swan 71 human trophoblast cell line, created spheroids, and then we can take the spheroids, encapsulate them in hydrogels, and then monitor their invasiveness or their motility over time. And this allows us to ultimately use these types of systems to study various biomolecules or how the cells are reacting to being in a three-dimensional network and how that affects their motility over time. So for the sake of time, I'm not going to go into all the studies, but they are cited in my talk if you're interested in learning more. But these are the types of things and output methods we can do with these trophoblast motility types of studies. We can look at nascent protein deposition by the cells to see what they're depositing within the matrix and how that might relate to various cell behavior. We've also used these models in particular to study different biomolecules and how they affect um, the trophoblast. So we were very interested in the stress hormone cortisol and how that would affect the trophoblast cells and their motility. So we did a lot of work looking at how cortisol affects motility of trophoblast cells. And we saw a decrease in the outgrowth area of the trophoblast over time in the presence of the stress hormone cortisol. And then finally, our ongoing work with this model system in particular is to determine the role of the ECM on trophoblast motility within the hydrogels and how various ECM factors such as composition, stiffness, etc., affect trophoblast motility. So that was one brief vignette. And in the remaining time, I will share one more, and I will talk to you about how we then use these hydrogel systems to create a tissue engineered model of the vagina. And so we were very interested in creating a tissue engineered model of the vagina, largely for understanding vaginal biomechanics. I have interests in childbirth injury and understanding how the biomechanics of the tissue affects childbirth injury, such as vaginal tearing. And also, the longer term goal of this project is for regenerative medicine approaches to creating implantable vaginal systems. And so overall, again, what we do when we create these tissue engineered systems is we take these complicated vaginal or tissue microenvironments in particular. So this is the one of the vaginal microenvironment, for example. And then from this, we determine what engineering criteria we want to incorporate into our model systems, and then begin to build these modular types of systems with increasing complexity. So for example, I will show you on the next slide how we take this complicated four-layer tissue and how we simplify it for an engineering model system. And so for us, we defined 5 different sets of properties that we thought would be relevant for a tissue engineered model of the vagina. So the first was mechanical properties, making sure we're replicating the stiffness as well as the strength and toughness of the tissue. Physical properties, such as the anisotropy, so like the directionality of fibers within the vagina that give it some innate biomechanical properties. Eventually replicating tissue architecture. So if you think about the vagina, it's a fibromuscular tube. So if we're building models of increasing size and complexity, thinking about how we can incorporate larger tube-like structures for these types of systems. Again, replicating relevant cellular constituents. So to start, we'll talk about how we focus mostly on these vaginal epithelial cells, but eventually we would like to move into more complicated cellular sources as well. And then finally, replicating various components of the vaginal ECM. So that in this case, it's fiber components such as collagen and elastin. And again, in terms of the basic science part of things, we're interested in understanding how the vaginal microstructure influences the macromechanical properties of the tissue. And that's why with our tissue engineering model systems, we're not just focusing on the tissue, but we're going a couple length scales under that as well to the cells as well as the ECM components. And so I will now tell you a little bit about how we modified our Gelame hydrogels to be able to replicate more of the vaginal microenvironment. So I've told you a few different vignettes. We talked about using the base hydrogel, what we can do with it. I told you about how we can coat our hydrogels with ECM as well, especially if we're building stratified structures where we want cells on top of the hydrogels. And now we're going to talk a bit about hydrogel composites. So how you can combine hydrogels with other materials to create more complicated structures. So Gelame hydrogels and hydrogels in general are great for a wide variety of applications, but unfortunately, they're very soft. So as I mentioned before, we love Gelame when we're talking about the endometrium because it's approximately 100 pascals in stiffness, which is what the stiffness of the endometrium is. But if you know anything about the vagina, it's a much stiffer tissue on the scale of megapascals, not pascals. So we needed a way to reinforce the strength of our hydrogels to make them more physiologically relevant to the vagina. And so one way you can do this is to create fiber reinforced composites. So what we can do is we can take electrospun fibers and then we can use a dip coat method to create these fiber reinforced gels, and doing so confers a significant increase in the stiffness and stability of your hydrogel system over time. And so for us, what we did was we took our Gelme hydrogels and then we created elastin electrospun fibers because, again, elastin is a primary component of the vaginal ECM. And then we were able to create these fiber reinforced composites shown here. And this is showing the stiffness of our fiber reinforced composites. And we've shown that, the base hydrogel systems are approximately 100 pascals, but we can significantly increase the stiffness of our fiber reinforced composites to get much closer to that of vaginal tissue, which the stiffness ranges in the literature, but it's roughly in the megapascal range. The vaginal microenvironment is also in a very low pH. A pH of 4 approximately is what the vaginal pH is. So something else we wanted to do was to verify that we were able to use these types of biomaterials at a low pH. So from the literature, we used a vaginal fluid simulant of pH 4.2 to then test the degradability of these different biomaterials. So we used homogeneous hydrogels, fibers alone, and our fiber reinforced composites. And as you can see from this plot here, the fiber reinforced composites have the lowest change in mass over time, meaning that they are the most stable in the presence of a low pH environment, which renders them pretty useful for these types of applications. And then finally, we wanted to test the biocompatibility and cellular response of these materials because with the development of these new materials, we want to make sure that they are still useful in regards to cell culture in terms of a tissue engineered system. So we took primary human vaginal epithelial cells, encapsulated them in the fiber reinforced composites, and looked at viability, nascent protein deposition, and cell composite interactions. And what we found was that we had very high cell viability, as indicated by live dead staining. We demonstrated that the cells were able to deposit nascent proteins within the fiber reinforced composites. And then we also demonstrate by this SEM image that we're beginning to see cells beginning to spread in the materials as well. So now I've shared two different vignettes of how we create and approach these engineered systems. And now in the last couple minutes of my talk, I will share with you a few of the applications. So the creation of these engineered systems are great, but what do we do with them? And now we're starting to get into more technology that is more relevant to human physiology. So I spent a lot of time talking about Project Area 1, how we develop these systems to study pregnancy in humans and how we want to use them for drug delivery applications eventually. Project Area 2 in my lab is taking these types of systems and looking at space hazard effects on female reproduction. So largely we're focusing on low gravity effects on endometrial cultures in particular, which I will talk about on the next slide, but we're also moving towards space radiation effects as well. And then finally, we're working on developing organ chips to target urinary incontinence in older females as well, so we can learn how to better target incontinence and develop better therapeutics for that as well. So just, some brief preliminary data on what we're doing. I'm again really interested in space hazard effects on female reproduction, especially as we think about upcoming Artemis missions and missions toward Mars. Humans will be spending much longer time in space, and it's therefore relevant to understand how it's affecting their reproduction. So we can use ground-based systems to study aspects of space travel on the Earth so we can infer what is going to happen to astronauts in space. So what we're doing is we purchased this rotating cell culture system device, which rotates our samples in free fall to mimic low gravity as you would experience in space. And we can put our hydrogel systems within these chambers and then look at how the cells are responding to this lower gravity um, environment. And so far, this is just showing data from one experiment, but we started looking at the differentiation effects on endometrial stromal cells and if that's impacted by being in microgravity. And our preliminary data is showing some interesting results that do suggest that microgravity does affect the cell's ability to undergo this differentiation response during the menstrual cycle, which is relevant to pregnancy. So again, this is just one experiment, and we'll be repeating this later on to see if we are still, seeing this happen. But some exciting work and a demonstration of what we can do with these types of model systems. So to conclude my talk, I've talked about the biomaterials we use, how we use these biomaterials to create these tissue engineered models, and then also highlighted some example applications of what we're doing with them and our hopes to move toward translational science and creating more model systems that can be used from, from bench to bedside, essentially. If you're interested in women's health engineering, there are a few different opportunities I'd like to highlight. We are putting together the first book of its kind on biomaterials for women's health engineering. This is expected to come out sometime this year in 2026. We have 24 total chapters by experts in the field, and this book will again be the first of its kind to really summarize how biomaterials are used in the context of women's health engineering. So please check out this book once it comes out. And then if anyone else in the audience also does women's health research, we are soliciting submissions for a special edition collection in communications engineering. This special edition collection is entitled Technology for Modeling, Monitoring, and Maintaining Women's Health. And we really aim to showcase how engineering research can be used to, cover and study a wide variety of aspects of women's health. So this will also come out late 2026 or early 2027. So you can either submit something for this if this is also your field, or check out the collection once it is published. And so with that, I would just like to thank my team who have been instrumental in, doing all the work for our lab. And I would also like to thank and acknowledge our funding sources, especially the CCTS, who have funded us for these projects in particular. And with that, I am happy to answer any questions you might have. Thank you for your attention.
Oh, that was wonderful. Thank you so much. So we do have a little bit of time for questions. Feel free to either unmute, put your hands up. Looks like Victoria, I see your hand first. We'll give you the floor.
Oh, I was clapping. That's like a clap,
Oh,
like a congrats, not a
well, now you
question.
have to ask a question. Sorry.
No, that's totally fine. I guess okay, so I guess one question that I can ask, and it kind of builds on Maria's, which is like, do kind of take if you want to take these things into space, like are you planning to um look at how these kind of gels are compatible with like the different gravity and different, you know what I mean? Like taking them to these different spaces to make sure that they work. Because Maria's was about also making sure that the cells are differentiating properly in those spaces too. Because those processes may also be different, but also making sure your materials are still compatible with that.
Yes, great questions. I mean, so far we haven't had any issues with gels degrading or anything. But we do have a wide variety of different control experiments, ground-based control experiments planned to make sure that our systems can, effectively go up to the ISS, moon base, whatever situation we are at the time. So a lot of our work is proof of concept and verifying that, and also taking our current system and adapting it to be compatible to those types of missions. Because a lot of those missions, you really want to reduce the astronaut hands-on time. So you need to figure out a way to make it so that it's not so, um, so experimentally, involved for the astronauts. So we're moving to on a chip format, and we've also talked to Space Tango in town, who is an implementation partner, and they have, a Cube Lab set up where it's basically a standalone thing you can just plug into a space shuttle, and it will just run the experiment autonomously. So we are improving our technology to get to that point as well. So both good questions. Thank you.
I'll do another one just because. Um, so I know because I am on the TL1 and I've seen your grant. I know that you're adapting this to other organ systems like a bladder on a chip. Do
Mhm.
you foresee also creating like lungs? Because I feel like that's also kind of pertinent to space travel, kind of adapting it to like a whole body situation, or is it mostly just on women's reproductive organs at the time?
So as of now, we're mostly focusing on the female reproductive system as well as the female bladder because we're mostly interested in women's health applications. But I have had a collaborator approach me for a very exciting other project that I will keep secret for now. But another aspect that we think would be a really, really cool type of model system that can be expanded not only to women, but to other people as well. But it's still secret right now because I think it's a really cool idea. So stay tuned.
So I'll ask a question, so I don't know the difference in gravity between Moon and say Mars, which play the two different space directions, I guess, that we're going. So are you checking them against both, or are you mostly focused just on the Moon?
So right now we're focused on microgravity. So that's largely, moon and thinking about the space station because that's where most humans have been right now. And that's where most of the human data is from. Um, But there is also the option to start looking at hypergravity, so increased gravity, and that's relevant in terms of thinking about, space shuttle launches, other types of systems as well. So for us, we're mostly just focusing on the microgravity space because there's more data on that, um, especially with human cells, but there is an opportunity to go in that direction as well.
May I ask a question? Yes, thank you. Thank you, Samantha. It was really interesting for me. I just interested about in mathematical modeling.
Mhm.
Which mathematical model have you used? And including this previous question about the gravity, maybe we can make like a mathematical model which taking in account not only gravity but electric, magnetic fields, and so on and so on. So the many stuff can be included. So which mathematical model have you used? Yeah.
I have used none, but that sounds like an amazing collaboration. I have been looking for someone who can help with that. Because I have some really cool ideas of what we could do with like even like, FE modeling on like I know people who do FE modeling on like the uterus and like childbirth. So if we had the information, like the math there and the model for that of how the female uterus is experiencing forces, that would be really cool information. So if you're interested,
Samantha,
I would
maybe
love to chat.
yes, maybe can we meet later because of I have bunch of ideas, That's
Yes,
my initial specialty, this applied mathematics.
I
So
would love to talk.
yeah,
That we are definitely looking for someone.
yes,
So
my
I will
name is
definitely set that up
Fahik
with you.
Alipova. Yeah. Thank you. Yes. Sorry for time. Yeah.
All right, so very good questions there. Samantha, please take a look at the comments or the comment section. We get a chance. A lot of people are very appreciative of your talk today. I'll make sure you saw that. In the interest of time, I know we've got to everybody jump on to the next thing here. But thank you very much for getting the Spring Translational Science Seminar Series kicked off so well. Everyone have
Great.
a
Thank
wonderful day.
you.